So this lecture is just going to briefly go over metabolic energy. This is part of chapter 3, and we'll talk about it more in class on Tuesday. So what is the energy currency of the cell? Of course, we've all heard of ATP. That's what we know. This is considered where energy, how we get energy in the cell, how we use energy is usually in the form of ATP. So ATP, as we talked about, is a nucleotide. We don't think of it as such, but it is a nucleotide. So we have our nitrogen base, our 5-carbon sugar, and then we have 3 phosphates attached to ATP. And so that's where the triphosphate, adenosine triphosphate, tri from the 3 phosphates. And so what makes ATP such a good storage of energy, or such a good source of energy, is these phosphate bonds. So the bonds that are holding the phosphates together. So typically here's one phosphate connected to another. This bond is considered a very high energy bond. I mean, there's a lot of energy packed into this bond. So if we find a way to break this bond or hydrolyze ATP with water to release this phosphate bond, release a phosphate forming ADP, in the process of doing that, if we break that bond, we release that energy. And so the energy released from ATP can then be used to power other reactions inside the cell that require an input of energy. And so we can go from ATP to ADP by removing one phosphate, or we can go from ATP to AMP, adenosine monophosphate, by relieving 2 phosphates, which we also refer to as pyrophosphate. So that's going to release twice as much energy in the process. One thing to keep in mind when we're talking about the use of ATP, let's just remind ourselves about Gibbs free energy. So remember, that's delta G that we've talked about before, not in this class, but in other classes. So this is not a chemistry class, so I'm not going to ask you to actually remember the formula for delta G. But what I do want you to remember is that a positive delta G, so any reaction that has a positive delta G, that's considered not spontaneous. So if you have a positive delta G, that means that you have to input energy in order for the reaction to occur, which means that the final products of the reaction are going to have more energy in their bonds than are the reactants that produce those products. And negative delta G is exactly the opposite. If you have a negative delta G, that's a spontaneous reaction that will occur naturally in the cell. All right, so let's look at how ATP is able to drive unfavorable reactions, why it's the energy currency. So we'll start with glycolysis, which we'll talk about in a little more detail in a second here. But glycolysis, the first step in glycolysis is to convert glucose to glucose 6-phosphate. This is considered an unfavorable reaction when we have a positive delta G of 3.3 kilocalories per mole. And so what makes it a positive and unfavorable reaction is that this glucose 6-phosphate, there is a more energy present in the bonds of this molecule with a phosphate added to it than there is in glucose alone. So by adding this phosphate to it, we have higher energy in this molecule. So in order to produce a molecule that has higher energy in its bonds, we have to input energy for it to occur. So that's why it's a positive delta G. This isn't going to occur naturally in the cell unless you have some sort of energy coming from elsewhere. So again, the glucose with the phosphate, still glucose 6-phosphate plus water, this is going to be considered a high unfavorable reaction. So we can couple this reaction to ATP hydrolysis, where we can hydrolyze ATP at the same time as this reaction. So we hydrolyze ATP, that's going to remove the phosphate from ATP. This phosphate can then be transferred to glucose to form glucose 6-phosphate. Now when we hydrolyze the phosphate from ATP, that has a delta G of negative 7.3 kcal. That's a favorable delta G. So since we are releasing more energy here with the hydrolysis of ATP, then we are requiring to input to add a phosphate to glucose. This is considered a reaction. We can use the energy from ATP to power this unfavorable reaction here. And so as a result, when we couple the reactions together, our overall delta G is negative 4 kcals per mole. Take negative 7.3, add 3.3. And that's going to give us our total, because it's a negative value, that tells us this reaction can occur if we couple it together. So this is how we use ATP to drive the unfavorable reactions inside the cell. Okay, let's look at glycolysis in a little more detail on how we're getting ATP from glucose. So the very first step in metabolism for most cells is going to be breaking down glucose to get ATP through the process of glycolysis. All right, so talk about glycolysis. So here's the stage of glycolysis, and I don't need you to memorize glycolysis. I don't want you to remember all the intermediate reactions in glycolysis. I'm sure you can do that in biochemistry if you want to. You don't have to do that in this class, but what I want you to know for glycolysis and all the reactions that we talk about is what goes into the reaction, what comes out of the reaction, and then what you get along the way. So what you start with in glycolysis, we're going to start with glucose, what you're going to end up with, which in glycolysis will be 2 pyruvates, and then how many ATPs and other molecules are you getting along the way. So for glycolysis, we're going to start with glucose. We're going to convert that to pyruvate, actually 2 pyruvates. And in the process, we're going to yield 2 ATP and 2 NADHs, which are important for electron carriers. We'll talk about what that means. So that's basically what I need to know for glycolysis. Now, what you should know in glycolysis is there are really 2 steps. There's the first step, which is the energy consumption phase. So the beginning of glycolysis is you have to actually put in energy. So this is showing the energy consumption phase, starting with glucose and then going down to fructose 1,6-bisphosphate. You can see we're going to invest 2 molecules of ATP from the start. And at the very beginning, we have to invest a molecule of ATP. We talked about this reaction just a minute ago. And then we're going to invest another molecule of ATP here. And so there's an initial investment of energy before you can then get the energy out in the end. As a result, this means that glycolysis is going to be considered a regulated process. Meaning because we have to invest energy for glycolysis to occur, we only want to actually go through glycolysis when we need to. So the cell's only going to go through glycolysis when it actually has a need for energy. So the cell is going to regulate glycolysis at 2 enzymes. So the hexokinase, which is important for the first step in glycolysis, and then phosphofructokinase. And so remember kinases? I have our kinases that always when you see a kinase, you know you're phosphorylating. So look at our 2 reactions. Our kinase is phosphorylating glucose to glucose 6-phosphate. Same thing here. Our phosphofructokinase also phosphorylating fructose 6-phosphate to fructose 1,6-bisphosphate. All right, so we want to regulate glycolysis, that we only go through it when we actually need to go through glycolysis. And so how that works is if we have a high amount of ATP in the cell, so if we have a lot of ATP, meaning we have a lot of energy around, we don't really need to make more ATP. So that's going to be a signal to the cell. If we have ATP, then let's kind of slow up on this glycolysis thing. No need to go making more ATP when we already have a sufficient amount. So if there's a lot of ATP around, ATP in high concentrations is actually going to inhibit the activity of phosphofructokinase. So high ATP means that this enzyme here is going to be inhibited. Now, if phosphofructokinase here is inhibited, if it's not functioning, that's going to lead to a buildup of the molecules ahead of phosphofructokinase in this pathway. So if we don't have a functioning phosphofructokinase, we're going to have a buildup of glucose 6-phosphate. We'll also have fructose 6-phosphate, but these 2 molecules interchange pretty regularly. So then we're going to have a buildup of this glucose 6-phosphate concentration. So our concentration of glucose 6-phosphate is going to increase. And as a result of that, now we're messing with the chemical equilibrium of this first reaction here. So remember, enzymes are great, but they're not miracle workers. They cannot make a reaction occur against its chemical equilibrium. So if we have a high concentration of glucose 6-phosphate, that's going to decrease the activity of hexokinase because this enzyme is not going to want to convert glucose to glucose 6-phosphate when the equilibrium of this glucose 6-phosphate is, or the concentration of glucose 6-phosphate is much higher than glucose. So we're going to have an inhibition of this enzyme of hexokinase, and as a result of that, then we're going to have an increase of glucose concentration. This glucose concentration, if it's not going to be broken down by ATP, will likely be stored in the form of glycogen. Okay, so that takes us through the ATP investment stage. Now let's talk about where we're getting ATP. So how that investment of initial energy is going to actually lead to more energy in the end. So here's our ATP generation part. So we start with our fructose 1,6-bisphosphate here that we got from the previous reactions. And this is going to get split into 2 molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And then so the dihydroxyacetone phosphate will actually be converted to glyceraldehyde 3-phosphate. And then both of these are going to go through our energy production phase. So now we're going to have, you see how we have kind of this shaded region where we have the darker and the lighter. This is just showing that everything going through here through the pathway is actually done in duplicate. So we're going to have 2 3-carbon glyceraldehyde 3-phosphates that came from our 6-carbon glucose that are going to go through the energy production phase. And so this part here, now we're going to actually end up producing, so we're going to produce ATP. And the reason why we can produce ATP is that breaking certain bonds, there are certain bonds in the cell that actually have a higher energy in them than ATP phosphate bonds do. So if we break those higher energy bonds, we can actually produce ATP from those. So that's going to happen in 2 steps in this process. So we're going to have a production of ATP here, and then we're going to have production of ATP elsewhere. I'll show you in a second. We're also going to produce NADH. So if we look at here, we're going to produce NADH at this stage here, actually 2 NADH. All right, so let's go down to our 2-phosphoglycerate, then we'll continue on. So here's our 2-phosphoglycerate. I apologize for the fuzziness of this particular image. This 2-phosphoglycerate is going to be converted again through a series of steps, and then we're going to actually produce 2 more ATP here to end up producing pyruvate. So you can see in the second half of the reaction, we produced a total of 4 ATP. But because we had to invest 2 ATP at the beginning, we get a total net gain of 2 ATP from glycolysis. And then we also had the 2 NADH. Don't forget about those because those will be important. Okay, so all this process of producing pyruvate from glucose occurs in the cytosol. Now, this can happen both aerobically and anaerobically, so with or without oxygen. In cells that are growing aerobically with oxygen, this pyruvate is then going to go to the citric acid cycle, and this will take place in the mitochondria. Cells that are growing anaerobically, this pyruvate is actually going to be broken down into two molecules. It's actually going to be converted to one of two ways. It can be converted into lactate, and this is going to use the NADH that was generated previously, or it can be converted to acetaldehyde and eventually to ethanol. And again, it's going to use the NADH that was also produced previously. So we'll talk more about that in class and what happens with that. But let's move on and talk about cells that are growing aerobically, since that is the majority of cells in our. All right, so oxidative metabolism, those that are growing aerobically. So we'll take the pyruvate that we got from glycolysis, and the first step in oxidative metabolism is that pyruvate is going to be converted to a molecule here. This CoA is known as acetyl-CoA. So this here is acetyl-CoA. And so when we do that, we're going to produce NADH in the process, another electron carrier, which will be important. All right, and so this acetyl-CoA that's produced, this is going to go through the citric acid cycle. And so remember, all of this pyruvate acetyl-CoA, this is now taking place inside the mitochondria. So here we are in the citric acid cycle. And so remember that since we had 2 pyruvate coming from glycolysis, we're going to have 2 acetyl-CoA. So for one molecule of glucose, those acetyl-CoA are going to go through the citric acid cycle. We're actually going to have 2 total turns of the cycle because we're coming from a 6-carbon molecule to the acetyl-CoA, which is actually now a 2-carbon molecule. So our 2-carbon acetyl-CoA is going to combine with oxaloacetate in the citric acid cycle to form citrate. And so then the citrate's going to go through the process. And again, I don't need you to memorize the steps of the citric acid cycle. I want you to just know what we're getting out. So if you go through and count along the way here, you'll see that we are going to get a total of 1, 2, 3 NADH. We're also going to get 1 GTP. Now GTP is functionally equivalent to ATP, so think of it as just getting another ATP. We're also going to get 1 FADH2. Also in the process, we're going to pick up 2 carbon dioxide. So this is how we're going to go from a 2-carbon to with a 4-carbon. So this is how by losing 2 carbon dioxide, this is how our 6-carbon citrate is then going to be converted after all to 4-carbon oxaloacetate. We're going to lose 2 carbons as carbon dioxide. All right. So keep in mind that, remember, 3 NADH, 1 GTP, which is equivalent to ATP, and 1 FADH2 from one turn of the citric acid cycle. Now, if we're going to figure out how much we're getting from a molecule of glucose, all of that is then going to be doubled. All right, so we've gotten through this process. We went through our TCA cycle, and if you count up how much we have from one molecule of glucose, we really don't have a lot of ATP. So we've gotten 2 ATP total from glycolysis, and then we've gotten another 2 ATP from going through the citric acid cycle twice. Okay, that's 4 ATP from one molecule of glucose. It really doesn't seem like we're getting that far. So where a lot of the energy is actually going to come from in oxidative metabolism is the NADH and the FADH2. So where do those go after they're produced? Because we produce quite a bit of those. So where are those going to go? And that's where we get into the concept of the electron transport chain. So our NADH and our FADH2 here are known as electron carriers. And so they're actually going to carry electrons to, this is all taking place in the inner membrane of the mitochondria, and I'll show a better image that actually shows that to you. So we're going from our NADH with its electrons, those are going to be transferred to this electron transport chain. So in the inner membrane of the mitochondria, we have what are known as complexes. We have a total of 4 complexes. And so NADH is going to transfer its electrons to components in the complex. I'm not really interested in you memorizing the exact components in the process. I'm more interested in you knowing the process of the electron transport chain. So electrons are first going to be transferred to this FMN and FES. And in the process of transferring electrons to these, what's going to happen is protons are going to be pumped across the membrane, and eventually we're going to end up producing ATP. And I'll show a better image that makes it more clear. This is a little unclear here. So basically what happens in the electron transport chain is that molecules are going to transfer electrons to other molecules that want them more. So NADH will transfer electrons to FMN because it wants it more than NADH. Then FMN will transfer to FES. to CoQ, and then down the chain as it goes. And each time electrons get transferred to a new molecule, it's transferred to a molecule that wants it more, and as a result, it's an energetically favorable reaction. So we're going to release energy in the process of transferring electrons down the chain. Okay. So if you look, starting with NADH, going all the way down the electron transport chain until we finally transfer electrons to oxygen to form water. So oxygen is the molecule that wants electrons the most out of all here. So in the end, that's the final step, is converting electrons to oxygen to produce water. We're going to release 3 ATP from our molecule NADH. FADH2 comes in at a slightly different point, so FADH2 can't transfer electrons to this FMN. Instead, it transfers its electrons to FES, which goes to coenzyme Q. Since it comes in at a later point, we don't get quite as much ATP from FADH2. We're actually going to get a total of 2 ATP from a molecule FADH2. So if you look, each NADH that we've gotten through metabolism will actually be used to produce 3 ATP through the electron transport chain, and each FADH2 will be used to produce a total of 2 ATP through the electron transport chain. All right, and I want to show a better image that actually shows where this is. So if you look at this, we're actually showing that in the inner membrane of the mitochondria, this is where our complexes are going to be located. And this shows a better description of what's actually occurring here. So from our NADH and our FADH2, we're going to transfer electrons to each of the complexes. So NADH transfers electrons to our complex 1 here. And in the process, we're going to convert our NADH to NAD+, so we're going to lose our electrons. And as that's happening, this is an energetically favorable reaction. So as this is occurring, energy is released. And what this energy release does is it allows protons here to be pumped across the membrane of the cell. So we're going to be pumping protons across the membrane. This is going to take place here in complex 1, as well as complex 3 and complex 4. So those are the 3 areas where we're actually going to have protons pumped across the membrane. So what we've done now is we're creating a proton gradient. So we're creating where there's a larger concentration of protons here in what's known as the matrix, so the area between the 2 membranes of the mitochondria here in the matrix. So we're going to have a larger concentration of protons here between the 2 membranes. And we're also going to have, so we have a concentration gradient. We also have a charge gradient because we're pumping positively charged compounds. We have a higher concentration of positive charge. So we have more positive charges here and more negative charges inside the mitochondria, inside the inner membrane. So because we got this charge gradient, these protons that are then pumped across, eventually these are going to want to come. We want to establish equilibrium. So this proton gradient is actually going to pump, these protons are going to pump back into the cell through a molecule known as ATP synthase. And since we have this gradient built up, the pumping of protons, or the coming of protons back into the cell, this actually releases energy. And so the energy released in this process is going to be used to convert ADP to ATP. So we're going to release energy to produce ATP. And then this ATP, this is how we're getting our ATP from NADH and FADH2. So based on the amount of protons that are pumped across for NADH, we're going to pump enough protons across the membrane that we're going to produce a total of 3 molecules of ATP. And from FADH2, we're going to pump enough protons across to produce 2 molecules of ATP. So that tells us how we get ATP from glucose. But of course, glucose is not the only molecule the cell can use to actually grow and metabolize with. So let's talk about other molecules. So there are other sugars that can be used. Glucose is the main one through glycolysis, but other sugars can definitely be used. It's not like every sugar we eat always involves glucose. So other types of sugars that can be used, those are going to be converted either to glucose or to some other stage in glycolysis. So a lot of other sugars can be converted to molecules that can enter glycolysis either at the beginning through glucose or elsewhere in order to obtain energy from them. Nucleotides also, remember, nucleotides contain sugar, so nucleotides can be converted into their sugar. The sugar portion of the nucleotide can also be used to generate ATP. We don't often break down nucleotides, but it can be done. Amino acids can also be used. Now, amino acids, we don't get as much energy from them because they don't go through the entire TCA cycle. The entire glycolysis through the citric acid cycle. What amino acids do is they're going to be converted to citric acid cycle intermediates, and there's a variety. Some converted to oxaloacetate, others to citrate. I'm not going to make sure you actually don't worry about knowing all those details. Just know that the amino acids converted to intermediates of the citric acid cycle, and they can be broken down, entered into the cycle, and be used to generate ATP and the electron carriers through that process. Now, lipids, this is where we can get a lot of energy and from a very small molecule, really. So remember, lipids are composed of the triacylglycerols, where we have the 3 fatty acids connected to glycerol. And so the glycerol portion of this can actually be converted to a molecule that goes into glycolysis. So glycerol can go into glycolysis. Glycerol is really a sugar intermediate. And then the fatty acids from this portion go through their own oxidative process. So let's look at an example of that. Here we have a fatty acid that has 16 carbons on it. So we have 14 repeats of the CH2 plus 1 carbon here and 1 carbon here. So we have a 16 carbon fatty acid with its carboxyl group on the end. What happens is we're going to convert that 16 carbon fatty acid, we're going to add that to acetyl-CoA to produce a 16 carbon acyl-CoA. And the process of adding acetyl-CoA to this, we're going to have to invest ATP, because we actually have to, this is not a favorable reaction, we have to add a little ATP for this process to occur. So we do lose a molecule of ATP in the process, but don't worry, we're going to get so much back in the end, it's going to be ridiculous. All right, so here we have our 16 acyl-CoA. And then what happens is that this 16 carbon acyl-CoA is going to be broken down in a stepwise fashion. We're basically going to take 2 carbons off at a time, combine that with the CoA to form acetyl-CoA. So we're basically going to take this 16 carbon acyl-CoA. It's going to go through a reaction where we're going to produce FADH2 here. And that's going to form, and then so we have our FADH2, we release that, and then we're going to release an NADH. And in the process of this stepwise process, so don't worry about each individual step, just know we've produced 1 FADH2 and 1 NADH. We're then going to release our acetyl-CoA. So we took our 16 carbon acyl-CoA, we took off a acetyl-CoA from that, and what we're left with now is a 14 carbon acyl-CoA. And so this acetyl-CoA that we've released, this is going to go into the citric acid cycle. So we're going to go through the citric acid cycle through this. This 14 carbon one is then going to go through the same process. We're going to release 2 carbons from our 14. So we're going to go from 14 carbons down to 12 carbons, releasing another acetyl-CoA that goes through the process. And then that 12 carbon will go to 10 carbons, and then we'll keep going every time we release an acetyl-CoA. So if we started with a total of 16 carbons, that can be converted into a total of 16 divided by 2 is 8 acetyl-CoAs that go through the citric acid cycle. So we're getting all the things we get from the citric acid cycle 8 times. Additionally, every time we do this, we're also going to produce an NADH and an FADH2. So in class, we'll actually go through and do the calculations and figure out how much energy we're getting from one molecule, one fatty acid molecule. And you can see, so we can talk about how fatty acids relate to glucose in terms of how much more energy we're getting from a fatty acid compared to a glucose. And so we'll talk about that in class this week.